I'm going to blast till 2 o'clock and take a break like normal. To get through water systems, you have to get a little terminology. Oceanography is marine systems. Salt water, when I talk about oceanography, I'm talking about marine systems. I'm talking about salt water. I'm talking about something with salinity. That is oceanography. Limnology is freshwater, lakes, streams, creeks, anything that doesn't have salt water, basically. So inland, terrestrial, aquatic systems. That is your limnology. So limnology and oceanography, L&O, there's papers for this, there's meetings for this, there's places we go that have talks about limnology and oceanography. All right? That's the difference, that's the separation. Now, we've talked about some of this stuff here before, so this is not exactly new, but when you're dealing in the water column, and I say water column, when I mean water column, I mean from the sediment, from the bottom of the lake to the top of the water, or bottom of the seafloor to the top of the water. That's the water column, all right? So when you're in the water column, oxygen, obviously, we've talked about it 1,000 times, it's important. Temperature, is it cold, is it warm, does it change? Especially with oceanography, there's a lot of conditions and it's always 4 degrees Celsius. Bottom of the ocean is always 4 degrees. They don't care about temperature, it's always 4 degrees, always sucks. pH. pH ranges, you're in water. pH changes with water with the amount of hydrogen, with the amount of with the amount of dissolved substrate. The pH can change, either you like it or you don't. And then light penetration. Light only penetrates a certain distance through water. And so if you are photosynthetic, you better be near the top. If you're in Galveston Bay, you better be right on the very top because it doesn't penetrate much at all because it's chocolate milk water out there. All right? So depending on where you are, light penetration has a big issue. The two biggest gases to deal with in water situations, the same one you pretty much deal with in the air. Carbon dioxide and oxygen. Because obviously oxygen is there to breathe. Are you aerobic or are you anaerobic? We talked about those that are anaerobic and aerobic. We talked about photosynthesizers that were both aerobic, oxygenic, and anoxygenic photosynthesizers. So oxygen is important. Carbon dioxide, not only is it there because or important for primary producers. Remember, primary producers take CO2 and convert CO2 into organic matter. But also carbon dioxide has a big issue with pH. You increase the amount of CO2 that is dissolved in water and you lower pH. It becomes more acidic. We talked about that with global warming. We talked about that with CO2. We talked about how the corals, if the CO2 concentrations get too high, the pH of the ocean goes down, the corals go extinct, so CO2 has an issue. The other ones are more specific to different lineages. Nitrogen, if you're dealing with nitrogen, if you are cyanobacteria, you're fixing nitrogen into to ammonia. If you are a nitrate reducer, you're sitting beside a golf course, the golf course produces or dumps a lot of fertilizer, throws a lot of nitrates, you use that nitrate, you reduce the nitrate, and you breathe. So specific to you, hydrogen can also be an energy source. Hydrogen can be used as an electron donor. You can be an autotroph at this point by utilizing hydrogen. You can also be an autotroph utilizing methane. You oxidize that methane, CH4 to CO2, you are oxidizing methane, you are getting energy as an electron donor. So depending on the other gases, and there's some others in there, hydrogen sulfide is a gas, that will be on the list. There's some other ones on it, but the main two big ones are oxygen and carbon dioxide, and this is what I'm talking about with oxygen here. Temperature has an issue with oxygen. How does temperature affect dissolved gases? If it gets hotter, do you have more dissolved gases or less? More. Less. What happens when something boils? Yeah, that's the key to think about with that. Remember, if you have that crossed up in your head, think about boiling and you see bubbles and the bubbles come out. Because the hotter you are, the less gases are dissolved, which means you heat up water, you lose oxygen.
And cold water is more nutrient rich.
Cold water is more nutrient rich, absolutely. You get more stuff in the cold water. So, oxygen concentrations at depth, which is something that people don't realize, is there's an oxygen minimum zone in the ocean. The oxygen profile within the oceans, and typically more with the oceans. If you take oxygen concentrations, this is depth, so this is going down in the ocean. This is higher concentrations of O2, and this is your depth. All right, say in meters. All right, oxygen concentrations will spike and then fall with depth. At some point, they actually return back up to near what is up at the surface. And that is because the bottom water is being colder, has more oxygen. The bottom waters having more oxygen. Also, the bottom waters do have a lot of nutrients. There's also less organisms, there's less activity, there's less respiration going on. So the bottom waters actually are more oxygen rich. So in the very deep ocean, you do have an oxygen concentration. The reason for that little bitty spike at the surface is that depending on your conditions, depending on where you are, you can actually get a pulse of oxygen right at the surface. What produces oxygen right at the surface?
Algae.
Algae. Why does algae produce oxygen?
They're
They're
photosynthetic.
photosynthesizers and they produce oxygen. So depending on your situation, depending on where you are, you can actually see a small rise in oxygen just below the surface. And if you're more turbid, there's the two conditions that may or may not happen. But photosynthesis does produce oxygen right on top wall. So that's that setup. So CO2, this is what I was just talking about. I said it without having a slide up here. It has a pH issue on it. Now in oceanography and in ocean systems, in marine systems, you do have a carbonate buffering system. And what happens with a carbonate buffering system is, if you add more CO2, if you lower, I'm sorry, let me take that back. If you lower the pH of the ocean, carbonate starts to take up the hydrogens. Takes the hydrogens away, makes carboxylic acid, pulls the hydrogens out of the system, and will actually start to raise the pH. If you start taking away pH, if you start rising the pH, then carbonate starts to dissociate and you start dropping H's. So it's this carbonate buffering system within the oceans that keep the oceans at a pH that is, what's the pH of the oceans? Do you all know? 8.1. Thank you very much, Mr. Root. 8.1 is the pH of the oceans. What's the pH of your blood? 8.1. Yeah. So pH of the ocean is 8. It typically stays at 8, but like any buffered system, you can overrun the buffer. And so that's why with global warming and whatnot, we're worried that we're going to override that buffering system and then we'll start dropping the pH of the oceans. But it's a carbonate buffering system. Now, the good thing about pH and the good thing about having carbon dioxide in these waters is that you can create carbonate shells. You can have calcareous organisms like this coccolithophore here. This dude here has these little shells on it, these little shields on it. This right here is a carbonate. It takes the CO2, it takes the carbonate, it makes self. It makes its own structure. Like we have calcium carbonate in our bones. This takes calcium carbonate here and makes itself. These guys, coccolithophores, they are chalk. White cliffs of Dover are those guys right there, coccolithophores. It makes chalk. So when you're using non-synthetic chalk, if you're using real chalk, you're actually writing on the board with microorganisms. Freaky. So yeah, but that is these guys. Now, at higher pHs, if you get the pHs too high, these guys dissolve. And these guys dissolve with pH. There's a level within the ocean that's called the carbonate compensation depth, and is a depth where the pH starts to rise. Down at the deep ocean, where you dissolve more gases, not only do you have a higher amount of oxygen, but you also have a higher amount of CO2. The deep ocean, below about 3,000 meters, 3,500 meters, down to 4,000, depending on the ocean you're in, and depending on the point through Earth's past, this carbonate compensation depth, the CCD, changes. And it changes so that these little dudes dissolve when they get deep. Which means, in oceanography, wherever these guys have accumulated, and have accumulated in the past, and you drill and you go sediment record and you detect these guys, you can detect changes, rises, and falls of sea level. And we equate rises and changes of sea level with rises and changes of ice on the planet. You can get a rough estimate of global climate change, temperature changes, based simply on the presence or absence of these guys through the sediment record. It's pretty slow. If you know your chemistry, you know the history of it, and you can all go all the way back and do what's called paleoecology or paleoclimate analysis. It's pretty cool because calcium carbonate dissolves at low pHs, which is what this is talking about.
How well do those guys work for Harvard?
That's what it is, man, it's chalk. Yeah, right. It's the same thing. But yeah, that's it, that's chalk. So, with that, you do have different environments, and today we're talking about environments. Today we're talking about putting microorganisms into context. With marine systems, you have 3 different main zones in a marine system. You have the coastal environments. Coastal environments give you estuaries and salt marshes. Galveston Bay is an estuary, right? Laguna Madre is an estuary. Anything near shore is an estuary. Salt marshes, you go out to Seabrook, you can see, as you go over the bridge over to Kemah, you can see, if you're heading south into Kemah, if you look to your left and right, you see salt marshes, right? Those are marine aquatic systems. Low wave action until a big name storm rolls through. Typically low wave action, high nutrients, high activity, biological activity. Those are coastal environments. Open ocean. Once when you get offshore, water conditions change. The color of the ocean changes. Even the Gulf of Mexico turns blue. Oddly enough, it does turn blue. You get far enough away from Galveston, it turns blue.
I'll believe it.
So yeah, you have to believe it to see it. Or see it to believe it. No way around. It's been a long weekend. You guys don't understand what a weekend I've had. Oh God, I wish it was just that, man. So the meeting yesterday was rough. Open ocean. When you get out in the open ocean, the biggest problem with open ocean is nutrients go bye-bye. Most nutrients and most of what organisms, especially microorganisms, need to live, thrive, and survive come as runoff from the land. You get far enough out, and even rivers as dirty and nasty as the Hudson River, it turns blue off of New York. Because all those nutrients either settle to the bottom, to the benthic environment. Benthic is bottom, right? Benthic is bottom. It either all deposits, deposition to a benthic environment, or it's consumed by those more coastal, estuary, coastal environments. All right? So there are very different provinces out there. Here's the estuaries. Now, I could really talk about crossing up 2 classes. I'm not going to hammer you guys on oceanography on this. I will try like y'all to lessen the questions on this. Because, yeah, there's like whole sections we would go through and whole chapters we would go through on this material here. I'm not going to hit you guys hard on that. Know that the estuaries are near shore. Know that the estuaries is main.
So you're basically saying that the nutrients gone makes the water blue?
Makes the water blue. So when you get down into the Caribbean, you get down into nice tropical waters, you can see Cayman Island. You can picture in your head, you have crystal clear waters. You can see down 200 meters. You can see down to the bottom when you're coming into port. There is barely any activity.
So nothing lives? I'm sorry, nothing?
Very little. Activity level is low because everything's been used. Primary production is based upon sunlight. Primary production is also based upon nutrients. If you're in a tropical environment that has a heck of a lot of sunlight, that means these microbes can go nuts. And the only thing that stops them is nutrients.
But isn't there a lot of nutrients in coral reefs?
There's not a lot of nutrients in coral reefs. There's a lot. Coral reefs have a really unique situation because coral reefs, if you have a lot of runoff from land, which provides nutrients, that runoff from land sediments, it's sedimentation, it deposits. Deposition of sediment material, particulate material, actually choke the polyps and kill off corals. So if you have too much sedimentation, you kill corals. If you have just the right amount, you have a little bit of nutrients coming in, you have a ton of sunlight, corals need sunlight. The zooxanthellae, which was question 30, 40 something, depending on which version of the test you're at. Zooxanthellae inside the corals are photosynthetic. They help make the organic matter. As long as you have a little bit of nutrients, right temperature, right light, they're good. As soon as something goes wrong, the zooxanthellae pack their bags and go away and the coral's toast. That's cool.
And it's warm water, so the oxygen levels are low.
Warm water, the oxygen levels are a little bit lower, but the good thing is the zooxanthellae are producing a little bit of oxygen that helps the corals out. So localized, they're okay with the oxygen, but you get too warm, you knock off the temperature, the zooxanthellae aren't happy, they go away, the coral bleaches, and the coral dies, and then the tourists go somewhere else.
Now, I don't mean to keep kicking a dead horse, but you're saying that you go there, you get a sample of water, you're not going to find life.
No. You're going to find life. You find life everywhere. I guarantee I can detect life in your own planet right now. But You have less activity, less production, less primary production in warm tropical waters. You go to the Arctic when the sun is up, and there is a ton more life. Perfect example of that is, well, where do the whales go?
Yeah, but how will you have life if there's no nutrients?
In
Or
warm
there's
water to
things
give
like
birth.
that?
You have
Water to
some
feed.
nutrients, it's lower, it's capped off. And in part, with nutrients, with depth, there's nutrients at depth. Where do the nutrients come from at depth?
It's like,
From above.
it's like.
From above.
Yeah, it's like right now in Galveston Bay, the water's clear. Like if you go.
There's a
in
clear
December-
spot in Galveston Bay.
If you go right now in December.
Sometimes a year.
Yeah, if you go right now in December and you go into the bay, you can see straight down to the bottom.
You get certain conditions where you can't do it.
Yeah.
On average, the nutrients in the deep water come from above. What nutrient, when you fertilize your lawn or whatever, what's part of that fertilizer? Food. What sinks? Food. So all the activity, the dead organisms, the organic matter that's produced up here, sinks as down here. So that's what part of the nutrients are down
Unless
here.
it floats.
Now, where you see the most activity on the planet, consistent activity, is where you are in the low latitudes. When I mean low latitudes, I mean close to zero, which means close to the equator. Where you have an upwelling environment, and I can get into a bunch of reasons you get upwellings, but we gotta get through the other slides. You get an upwelling environment where you pull deep water nutrients up to the surface where the sun's shining. If you don't have that flood up, if you don't have that upwelling, that sun, the primary producers, everybody at the top, burns through the nutrients at the top and reduces the amount of total activity. Is there life in tropical waters? Yes. Do you have higher cell counts? Do you have more activity in cold water? Yes. And in part, that's the reason why when you get to tropical waters and you want to go look at the corals and you want to go see everything, the water's clear because there's not much going on. Now, there are individual isolated incidents that provide nutrients, that provide blooms, that make that area go crazy. And that's where like an upwelling environment gives you a nice green look. If somebody in here has on the phone, if you want to pull your phone out, look at something other than Facebook during class, I appreciate it. But if you pull up your phone and you pull up a chlorophyll A map, chlorophyll A is the, obviously you guys know now, should know, chlorophyll is the photosynthetic bit, that photosynthetic organelle that allows photosynthesizers to produce organic carbon. If you look at that, one thing that you will see, and you look at, and it's called C-WIFS, is the name of the thing. I think it's C, and it's W-I-F-S, I think is how you spell it. If you look at the C-WIFS data of the planet, and you look at, God, here's where I'm gonna get, West Coast, here, US, Texas, whatever, here, Asia on this side, Australia down here. If you look at that data, what you will see is this green tongue that comes off of South America that goes basically with the equator. And that's because based upon the gyre system, based upon the way the oceans circulate, based upon wind patterns and 12 other things, you get a localized upwelling here in the middle. The localized upwelling in the middle provides nutrients to the upper waters, which provide nutrients for photosynthesis, which produces a lot of activity. And that's because you gotta get nutrients to the surface. So, 20-minute bit on a 10-minute subject, but look it up, you'll see. But anyways, off of all of that stuff, with the whole thing with the estuaries have nutrients because they're near land. Nutrients are runoff off of land. The other problem with being here is that you have to be halotolerant. Because what if, like in the past week, when we've had ridiculous amounts of rain and you're sitting in Galveston Bay, salinity in Galveston Bay, salinity in the estuary, salinity in Clear Lake, salinity around here falls. Well, in the springtime, if you're a fisherman anywhere around here, you know the winds blow hard off the Gulf for most of the spring, which blows the Gulf water right into Galveston, which raises the salinity. And so now if you're living in this environment, you better be halotolerant because now you're high salinity or low salinity and it can change with tides. If you're offshore, salinity never changes. If you're near shore, salinity changes, so you better be halotolerant. Tidal mixing is what causes a lot of issues. It changes temperatures, it changes oxygen, and it changes salinity. So this is very much of a mixing environment. The other problem with this environment is not only do you get nice nutrients, you get stuff that you enjoy and stuff you like, but you also get a lot of pollution. All right, that's a lot of the other stuff that runs off into these areas. Now, the estuaries work as a very good biofilter for the open ocean, because what we see as pollutants, there are many organisms that can break down these compounds and help clean the waters before they get to the open ocean, which is one of the reasons why everybody says protect our estuaries, because these estuaries help degrade a lot of pollutants. Now, at the same time, you dump a lot of nitrate in these environments, and you can cause some things to get way too happy that you don't want happy. All right, like harmful algal blooms. You give these guys a lot of nitrogen, a lot of nitrates. These guys can bloom and they ruin your day. Here's some pictures of some of these. We talked about red tides before. We talked about them in terms of Florida, in terms of some work being done in Corpus Christi Bay area. These guys, like I said before, if you're ever in these areas, they give off some nasty, nasty compounds. There's some more stuff here. Brevetoxin is one of the more harmful algal blooms. Brevetoxin, these guys, it can kill fish. It can make the seafood not consumable. The Food and Drug Administration will come down and shut down beaches and shut down fisheries, waterways, everything if they have too much of this. Listeria is another big, nasty one. There's a story about, is it Listeria also, it says Chesapeake Bay in the south. Listeria actually also infects a lot of the waters, and I'm from North Carolina originally. The Neuse River and a few other rivers in North Carolina, because North Carolina has a lot of hog products, hog waste, barbecue's awesome. The University of North Carolina State University, rather, was doing a lot of studies on Listeria and trying to understand the toxins involved with Listeria. They built a new building, science research building, that they house some of this research in. Now, NC State is known for engineering. If you want to do engineering, if you want to get a great engineering degree, you get NC State. It's a great place to do it. However, they screwed up, and they engineered the building such that the hoods that these guys were working on Listeria and the toxins, the airborne toxins produced by Listeria, were not ventilated properly, and they were actually venting into one guy's office that was on the other side of the building, and he spent a lot of time in his office. And after a number of years of being exposed to the toxin, he started developing massive neurological issues, massive neurological disease that was directly linked back to this, and it's linked back to his just consistent exposure to that toxin.
I remember something about rivers in Maryland being contaminated, and I think it was maybe something related to that.
It's probably Listeria. There's a lot, and this will shut down fisheries.
Mm-hmm.
And
like the fishermen were getting sick or something.
And the fish get sick, you eat too much of the fish, and you get sick, and so they'll put limits on, do not eat a lot of the fish here because of, sometimes it's the enterotoxins, the enterobacter that we talked about, the crab bugs, and some of them are harmful algal blooms. It's a Listeria is actually one of them that would do it, and will shut down beaches pretty fast. But yeah, so they actually had to have a lot of big lawsuits and stuff like this, and re-engineer the building. It was ironic that School for Engineering engineered the building wrong.
Did he come out okay? They got?
No, he actually had permanent damage. It was not a happy ending, sorry. But, yeah.
Apple.
I'm hoping you're talking about that. We didn't get it right. That stuff there. But yeah, it is nasty. But then moving from there, the salt marshes are, and knowing some of these lineages now, and knowing some of this setup, you still get some of the exact same bugs, the exact same lineages, the exact same processes that you can have within the sediments. You can see that here, that we've talked about before, like when I showed you last week, the oxidizers, the aerobes, the nitrogen utilizers, the iron utilizers, and everything. You can get that exact same setup in the salt marsh. You can get it still within the water column because you can have the muds obviously set up a distinct barrier. But because of these saltwater conditions in these marsh systems, you get low flow, very fine particulate sediments. You can actually get almost a mobile mud layer. If you've been diving before, or you go look over one of the bridges, you can see like sediment passing through. And so you can get some sediment-associated microbial processes and properties within the water column just because there's so many things suspended in that water column. And so you can get different processes up in the water, and obviously in the sediments. We'll talk about the sediments in a bit. But with that though, whether you are near shore, whether you are offshore, you do have what is considered to be the photic zone. And that photic zone is the area where you get the most photosynthesis is possible, is the photic zone. This changes, obviously, if you're in an estuary, if you're in Galveston, at some point, the photic zone may be 3 centimeters because it's so turbid. Turbid means things suspended in the water. It's so turbid that light doesn't penetrate. In the open ocean, in the tropical open ocean, where there's not a lot of activity, you can get light penetration down to about 200 meters deep. 200 meters being 650, 680 feet deep. You can get light penetration. Typically, in most open ocean conditions, the photic zone is the top 100 meters. Still a lot of water, right? But that's where you have, and in the photic zone, photosynthesis outpaces respiration. So you see here, autotrophs exceed respiration. Below that, and you have the photosynthetic compensation depth. You have the photosynthetic compensation depth, that's where photosynthesis and respiration even out. Below that, respiration stops. And it totally depends on where you are in the system as to where you are in the ocean marine system as to how deep that system is. So then, also, not only can you cycle not only can you do autotrophy, not only can you do photosynthesis, but in marine systems, nutrient cycling is very, very important. There is this thing called the Redfield ratio. How many people have heard of the Redfield ratio before? Everybody, all right, good, we won't have to cover it. Good. So, what this is the CNP. CNP, what's the C?
Carbon, nitrogen, phosphorus.
Thank you. CNP, carbon, nitrogen, phosphorus. It is a ratio at which optimal growth will be obtained. Now, the Redfield ratio is 106 to 16 to 1. C to N to P, 106 to 16 to 1, which means if you have 106, let's call it grams, of carbon, if you have 16 grams of nitrogen, if you have 1 gram of phosphorus, then you will have optimal growth. If you only, so 106 to 16 to 1. Now, you will see that number change. Depends on where you're on the planet, depends on who you're talking about. That number does vary a little bit, and actually you can put in about 6 other nutrients on there, down to iron and everything else, and really get this thing all the way worked out. If you had only 90 grams of carbon, what happens to your growth?
Slows down.
Slows down, why? What is it limited in? Carbon. It becomes carbon limited. If you have less than 16 of nitrogen, you are nitrogen limited, which means if you are doing agriculture, if you are trying to figure out why your crops aren't growing too well, you can go out and you can do a soil analysis. You can go out and do an atomic analysis of your soil and figure out which one of the nutrients you are lacking, which means do you go out and you throw ash, which adds carbon? Do you go out and throw nitrogen, because that always adds nitrogen? Or do you go out and add phosphates, because that's going to add phosphorus? Because if you are low in nitrogen and you go and throw a ton of candy bars out there, it's not going to help you because you're nitrogen limited. So if you know this, which also means when we're doing our culturing, if we want to enhance the growth of our cultures, we figure out which one they're limited in. Because giving them the one that they got doesn't help them much. You give them the one they don't have. And that will help increase your culturing efficiency. It will help your growth rates. That's why growth in the tropics is not limited by photosynthesis, by light. They got light.
Just out of curiosity, how do you know what the number is on an organism? You don't know what it is.
Typically, and the cool thing is that typically, and the reason why Redfield's name is known is typically it's that. And for most of life, and how they get that is cell structure and is the basic components of a cell. And if you take an atomic structure of the cell and you take the phospholipids, if you take the DNA, if you take the proteins, if you take everything else, it works out to have about this ratio. And that's why it's the perfect ratio. Right? Cool. So that's what culturing. And remember, when we talk about cultures, we talk about media, we talk about complex and defined media. Complex media means, defined media means you define each one of the bits in it. So if you're going to define a media, you probably want to know this. At least be over it and you realize that you can change something on that list and increase resource growth. Right?
So is that what primarily farmers and agricultural do to all their soil, and as well as people like the EPA to go to test the soils to find out what's going on?
And that's why somebody that comes and does that to you, they'll come by and say, hey, you need to drop phosphates on your land because you're not growing stuff because of the phosphorus and stuff.
And people are serious about growing flowers and greenhouses and stuff like that. They have their plant
Not just
food
that, but
ratios.
Just cities in general that try to plant parks and trees, and even in downtown.
Exactly. You add and you put the right groups in that balance that number.
So if one of those numbers change, they all should change, right?
Well, that's the thing. Well, it limits. And so this is a ratio, and that's why I said grams. So if I said gram, I could say milligrams. And so, but what they're going to do is they're going to use 106 atoms to 16 to 1. And they'll keep pulling out chunks like that until one of them is lacking. And then that becomes nitrogen limited. Or carbon or phosphorus or whatever. Iron is on there also, like I said, a few others. But with that, and with microorganisms, and with a water column, and with these marine systems, what you develop is what's considered a microbial loop, which has a better picture here. And what the microbial loop talks about is the exchange between dissolved, let me go back here, here's here. Dissolved organic matter, DOM, particulate organic matter, POM, and you can imagine what some of those would be. Loop, again. Particulate organic matter, dissolved organic matter, you take and you are able to produce a microbial loop from microbes producing the carbon, producing the organic nitrogen, producing life, and then decomposing life at the bottom. And so then with that, you start being able to talk about from dissolved organic matter, the microbes take it up, viruses, bacteria, ciliates, that's the eukaryotes, that's the protist, right? Take up that dissolved DOM, dissolved organic matter. From there, they get consumed, the primary consumers, secondary consumers, sushi eaters, right? These guys poop, dissolved organic matter, things dissolve back into the water column. Particular organic matter turns into dissolved organic matter, which is then taken back up by the microbes, which are converted back into organic matter, which is consumed, which is pooped, dissolves, comes to DOM, and it's microbial.
And that's how life is, around the coral life, and that's how they're
So
able
it's the
to.
same. So anyway, not just corals, but estuaries, coals,
How they do with the nutrients.
So this is, there's plenty of life, it's just that anything that's available is immediately taken up.
Once you have that proper ratio, what usually causes it to go out of the ratio? Is it usually like human activities?
Well, it can be. So like if you dump a lot of nitrogen in the area, then nitrogen becomes in excess, which means the other two become limited. So if your golf course dumps a lot of nitrogen and phosphates, the carbon may be limited, which means the only organism that can really survive in the environment is the one that produces their own carbon. Which is the reason why you dump nitrates and phosphates into the water column, the photosynthesizers, the algal blooms go nuts because they make their own organic matter. Which then means you have a lot of production here, primary producers, here, the photosynthesizers, which then eventually means there's a lot of these dead bodies or a lot of things to consume, which means the consumers are all, if you're a respirer and you're a consumer, you're breathing oxygen, which means now you have a lot to eat, which means you breathe a lot, which means you drive down the oxygen concentrations, which means the Chesapeake Bay goes anoxic. And now, crab cakes and football, and now you're going to football.
But can like manipulating that ratio like damage certain environments?
It absolutely can. Like I'm just saying right now, that if you increase this, you increase the activity, if you increase the amount of production in the area, now the whole water system goes anoxic, and organisms like crabs, oysters, things that are benthic, things that can't go run away or swim away, are dead because there's no oxygen.
Okay.
Very much damage.
They do have awesome crab cakes, though.
Amazing crab cakes.
Yes.
But so now, that's the coastal environment. The coastal environment has a lot of nutrients because the nutrients are coming in from the land. Their runoff, their terrestrial runoff is where these nutrients are coming from. Out in the open ocean, and you see the word here, pelagic. Pelagic just means open water, right? Pelagic water, pelagic, that's out in the open water system. It is considered oligotrophic. We've talked about oligotrophic before. What branch of the proteobacteria were considered best adapted for oligotrophic environments?
Alphas.
Very much. Alphas were the ones that were in here, which also means there's a lot of alphas in the open water. In the open ocean, you see alphas a lot out there. But so out here, you are limited in many different things. Out here, one of the things that you do have, and one thing that you need to have, is the carbon. You need to start your ecosystems. And so if you're producing a lot of carbon, if you have a lot of photosynthesis, you'll have a lot of total life. Because that's what bases your ecosystem off. They're the primary producers. If you don't have a lot of primary producers, you can't have a lot of consumers. So if there's a lot of photosynthesis, there's a lot more activity. So now they're dominant, but they control how big and how plentiful that environment's going to be. How much photosynthesis can you have while you're out there? So back into the photic zone. Back out here again. You're talking about now much more clear waters. You're talking about in waters that have the ability to transmit light. You have the ability to do photosynthesis on this. And that's where now that upper water, instead of having runoff supply nutrients, the photosynthesizers make organic matter. The cyanobacteria fix nitrogen, make ammonia, make proteins that have nitrogen in them. Well, they die. Dolphins die too. But they die.
No.
They don't.
They live forever.
They're magical creatures.
Flipping little dogs.
Like unicorns.
It's always flipping little dogs. But so these guys die, and that organic matter escapes the photic zone because of deposition. And that's what this meteoric looking thing here is. And it's called marine snow. Because if you're sitting on the bottom of the ocean, which I had the pleasure of doing, you sit on the bottom of the ocean and you can see, and it looks like snow. It's photosynthetic carbon, it's photosynthetic derived biomass that falls on the bottom, which also means the more photosynthesis at the top, the more life on the bottom. And there's a direct proportionality between the amount of benthic life with the amount of photosynthetic life, photic zone, and that is marine snow. Now, some snow, like you have a little flurries or you have a blizzard, when a dolphin or whale dies, that's a lot of organic matter hitting the bottom. There's some really cool documentaries about whale falls, about organisms that are attracted to large amounts of organic matter that all of a sudden appear on the bottom of the ocean, and the organisms that are attracted to those and how they find them. And look it up, YouTube. There's actually really cool stuff on whale falls.
You do some cool time lapses of what it looks like.
And you see the whale just get stripped down to bone
Yep.
over the course of that time lapse, and then even the bone gets taken out. Yeah, it's really cool stuff. But that's big time. That's blizzard. But for the most part, it's just marine snow. It's a little flurry on the bottom, and that's the organic matter dropping down. 90% of organic matter produced in the photic zone stays in the photic zone. Only 10% falls. 90% of organic matter that's in the photic zone stays in the photic zone and is recycled to the photic zone. 10% falls beneath. Of that 10% in the open ocean, only 1% actually reaches the bottom. And of that 1% of that actually, or 0.1% of the total organic matter actually gets buried and trapped within the sediments. So you have a big time reduction from what's in the surface.
So when you drill, you only find 0.1% of what's in there?
What's on top.
So as fish poop, other fish eat it before it gets to the bottom? I just,
Yeah.
I mean,
Yeah.
it's a good way to put it in
It
my
is,
mind.
it is, yeah.
Think about that the
Yeah,
next
they-
time you eat.
Fish gotta eat.
Yeah, there you go. So when someone says, I eat bigger crab than breakfast, yeah, they're actually eating crab for breakfast.
So 10% falls over the photic zone.
10% falls out of the photic zone into the A photic zone.
And 1% reaches the bottom, or
Reaches
1% of the 10%?
It's 1% of total.
Okay.
So 1% of total reaches the bottom, and 0.1% of total is buried. So basically, it's orders of magnitude is what you're talking about. But that's marine stone. And this is what it looks like. Here's a picture I was talking about before I was wanting to get to, but it's a couple slides away. So you see, CO2 goes in, and the reason why we're talking about CO2 is because CO2 is converted to organic matter, and if you do complete mineralization, if you take the organic matter, like we talked about a couple classes ago about the candy bar, turn it from candy bar to CO2, if you take that candy bar that's made here, and you convert it, complete mineralization, it goes back to CO2, and it leaves the system. That's the organic, that's the carbon going organic, back to inorganic. Autotrophy to respiration, it goes out. Some of that organic matter, 90% stays here, 10% falls. Consumed here, 1% of what's made here reaches the bottom, and 0.1 gets buried in the sediments. All right, that's free information outside the textbook for you enjoying. But that is the microbial loop. That's the carbon pump that pumps carbon from the surface down to the deep water. And that is primarily driven by microbes. Thank you very much. That's why they rule the planet. But here, and even here, it does say the 100 meters here. The average depth of the ocean is 3,800 meters. That's why they have 37 here. Is the average depth of the ocean. The Pacific is about 3,890. The Atlantic's about 3,850, and then back and forth. But for the most part, you're talking about 3,800 meters is the ocean depth. Average ocean depth. Which is awesome because if I just said the photic zone is only 100 meters, and I said the average depth of the ocean is 3,800 meters, and oceans cover 71% of the planet, are we in more of a lit planet or a dark planet? Or is it maybe? More of this planet, more life on this planet has never seen its sunlight. Never seen it. Majority of life on this planet doesn't even have sunlight.
Well, even land, the majority is underground.
Over 2/3 of the biomass on the planet is actually below the seafloor. 4 degrees, anoxic and dark. Average life on the planet is 4 degrees anoxic and dark. We are the extremophiles. We are the extremophiles. Because I sure as hell I'm not living in a dark place at 4 degrees. You can give me all the oxygen you want, but I'm not living there. Nope.
What if they got keep it?
Yeah, I don't care. I don't
Okay,
care.
little Chinese food.
Yeah, Aaron, they're not doing it, just keep going, that's why I'm in this. But this is the cycle, and this is what I've been talking about here, and this goes through it, and this does a little bit more detail with it. But this is the cycle, this is going through and cycling carbon through the upper water column. What's left here, it says even here, perhaps 1 to 15% at a 500 meter depth. That's what I was talking about before, the average depth of the ocean is much lower than that, so the number is much closer to 1%. So very little actually reaches the bottom. Most of what's produced in the ocean stays in the ocean. Very little actually gets deep, and most of that stays actually in the top. Which, and the point of this is that one of the experiments, I'm never going to finish all the stuff today because it's much cooler than we've been talking about, is that one of the experiments, we talk about the Brakefield ratio, is that in the open ocean, one of the limiting factors in the open ocean is iron. And so we said that can oceans be used to draw down CO2? And help with global climate issues, whether this southern, that southern, whatever. CO2 is rising. So can the oceans pull down CO2? Well, experiments were done with, let's seed the ocean with iron. Let's give these primary producers more iron because they're limited in iron. So there were experiments that were done in the 80s and 90s where you would go out into the tropic oceans and literally throw iron filings into the ocean. And it created cyanobacterial blooms. Awesome. Cyanobacteria consume CO2, produce organic matter. Organic matter now is no longer in the atmosphere and goes back into the oceans. But if 90% of that stays in the upper water column, you really haven't done much of anything. It stays in the water, it stays in the ocean, and it doesn't really fix anything. And so they stop throwing iron filings in the ocean. It wasn't that way. But that's the story behind the slide. But, nitrogen cycling out here, these puffs and tufts are what these guys are here. There are different organisms out there that we've talked about before that are able to not just cycle carbon out in the open ocean, but also cycling nitrogen through the open ocean. These guys can produce and utilize into nitrogen gas, atmospheric nitrogen, fixing nitrogen in an aerobic environment. This is one of the factors, though, that limits a lot of the life that's out there in the ability to get the nitrogen into the system. Cyanobacteria are a good one that do this. There are some other organisms that are capable of cycling nitrogen through, but nitrogen cycling, that nitrogen fixation pathway into ammonia. Nitrogen fixation is what we're talking about here. There is Animox, which can cycle and really change. We talked about Animox before. That's nitrite plus ammonia cycling to N2. That is one of the sets here. It doesn't make as much sense on this bit, but we're talking about more about nitrogen fixation. Nitrogen fixation limits life in the open oceans because unless you fix nitrogen, you don't have organic nitrogen, you don't have life. So those organisms that can fix nitrogen also, just like the carbon fixers, help limit the amount of life that's out there. Now, we talk about life when we talk about bacterial populations. Now, unfortunately, we talked about clades. We talked about alpha proteoses. We talked about rhizobiales. We talked about deltas. We talked about all these Latin names. I may or may not mention this. To get a Latin name to be called the sulfovibrio sulfuricans, E. coli, the key with getting named is you have to be cultured. If you don't have a cultured representative, you don't get a Latin name. So one of the more dominant groups on the planet, bacterial populations on the planet, is SAR11. SAR11 was found by what's his name? Craig Ventnor. He's got about a good name. This dude did genetics all through the 90s and 2000s by talking NSF and allowing him to sail on his yacht around the oceans and collect water samples and do some DNA analysis and ship it back to land. And so brother spent most of his time just sailing around on the boat. Like, man, I'm jealous. That would have been awesome. But one of the things that he found doing his sequencing of the oceans was this SAR11 and Sargasso Sea. Who knows what the Sargasso Sea is? Who has been to North Carolina, South Carolina, Georgia, and northern Florida? Have you been to the coast? You were in the Sargasso Sea. The Sargasso Sea is a circulation pattern off the North Carolina, South Carolina, Georgia, Florida coast that circles water off the Gulf Stream, goes up about off Cape Hatteras, and circles back down, coming in about Daytona Beach, Miami area, and cycles back, and it's a circulation body of water. It's a sea that's bounded by a current, and it's called the Sargasso Sea. Who knows what sargassum is? What did you got in sargassum? Seaweed. Thank you. It's that floating bunch ball, yellowish seaweed thing that has like the kind of tubey with the little leaves on it, little branches. That bunch is sargassum. Yeah, it has little air bladder things in it and stuff like this. It's those things. That's sargassum. And it came from, when the explorers came over, they actually saw enormous mats of this stuff and thought they had found land. Because in the middle of the Sargasso Sea, this stuff gets trapped in these eddies and forms huge, huge bunches out there. And so sargassum, thus the Sargasso Sea in that area. But when he was sailing through the Sargasso Sea, when he was basically left Daytona and several drinks behind, he went out here and collected water, and out there, 25% of the microbes he found were at SAR11, and no one's actually cultured the thing yet. And so therefore, it's still called SAR11. But it's one of the more dominant bugs to be found. It is found in coastal waters, it's found in fresh waters. You can read it here. It's really, really, really cool. They're photoheterotrophs, so photosynthesizing, but they use organic matter for energy. So they
Does it destroy because it overtakes everything?
sargassum. Yeah, it can clog up some areas.
Yeah.
Yeah, completely.
Because they said that's like the biggest problem is that they can't find a way to kill it.
It floats around. Yeah.
So you said there were photoheterotrophs and what? Did you say something more? Or did I just imagine that?
Photoheterotrophs.
Oh, then I hear you also have the photorhodopsin too. So the rhodopsin is now this is not for sargassum. This is for solid level. Sargassum is used for the
What's the difference between a sea and an ocean?
size.
Just
Size matters.
So the size of the sea, so the sea is larger than the ocean?
Small.
Smaller than the
Small.
ocean? So it's ocean, sea, river, or?
Bay, small bay.
Bay Sea Ocean.
Yeah.
You know, you got the cable and you got the pond.
Huddle up Bay Area.
Those are diving well. But microbes in the open ocean, you basically have, we've talked about this before, most of them are aerobic. Some are, you do have, even in the open ocean, depending on where you are, you have oxygen minimal zones. You can have anoxygenic phototropes out there. You don't have a lot of those. Lithoheterotrophy. Litho.
Inorganic.
Hetero.
They use organic matter for their carbon source.
Heterotrophy. So you do have a wide range of different bugs out there. It is diverse. It's not just photosynthesizers. It's not just one type. You do get a lot of different types of organisms out here in these waters. You do have viruses. Viruses typically, if I tell you that in open ocean waters, you typically have about 10 to the 7th cells per mil. Bacterial cells per mil, 10 to the 7th cells per mil. You typically have an order of magnitude more viruses. So you have 10 to the 8th viruses per milliliter of seawater. That's a lot of viruses out there. Are they life? We've talked about this before, but and it says here average density is this, but it's one more order of magnitude more dense than whatever the bacterial population is. So they're saying here 10.6, 10.7, that would be assuming 10.5 and 10.6 for bacteria, which honestly is a little bit low. It's one more order of magnitude higher than the bacterial population, which most abundant, it says depends on what side of the fence you're on. They are here. Part of what happens with viral populations and bacterial populations is that as bacterial populations increase, then all of a sudden viral load increases, which decreases bacterial populations, which decrease viral populations, which then increase the bacterial, which then decreases. The two of them do a little arms race back and forth, dependent on each other. Because remember, these viruses are mostly bacterial phages, which means they only attack bacteria, which means that they limit the bacterial population and go back and forth. So it switches, and you see increases and decreases between particulate organic matter, which would be dead bacterial cells in this case, to dissolved organic material, which means now there's a lot of bacteria in the other parts dissolved. So this can go back and forth depending on the viral load which is in the situation, which is what these slides here are talking about for you. But then we get through this real fast and we'll take a quick break. This is true. The largest bacterial biomass is below the sea. The largest bacterial population on the planet is subsea flora. There's more life below the sea than below the oceans than there is in the oceans, which blows people's minds, but it is absolutely true.
As far as like diversity as well?
Yes, I will say that. Your book probably doesn't know that yet, but yes, because that's what we're doing right now. We're going through looking at diversity in every bit of every lineage, every phyla, everything that we're studying in the surface world, we are finding in the subsurface. We're finding bacteria, we're finding archaea, and now, as you guys know from right upstairs, we also have fungi in the subsurface. So we have eukaryotes as well. Eukaryotic diversity is lower than the subsurface, flat out lower. Bacterial and archaeal diversity is just as diverse subsea flora as it is above the seafloor. So, with obvious lack of the photosynthesizers. However, there are actually cyanures in the teeth, which we're trying to explain. We'll talk about that later. The part of the issue with living subsea floor is, and your book talks about barophilic. Barobarometer, baropressurephilic loving, pressure-loving organisms. A lot of these organisms are and must be barophilic because of the ridiculous pressures with depth. For every 10 meters you go through the water column, you increase 1 atmosphere of pressure. So if you're talking about 3,800 meters of ocean, you're talking about 380 atmospheres of pressure just to get to the average ocean depth. You go beneath that, and you're still getting more and more pressure. So they need to be barophilic, which means culturing these organisms, some of them are barotolerant, which means they can live at the bottom of the ocean. They also be cultured up in the lab upstairs. Some of them are strict barophiles, which means unless we culture them under pressure, which you guys don't want us to culture under pressure because that creates one hell of a bomb in the building. Yeah, it's nasty, but definitely not letting you touch it. Yeah, you perk up on that one now. But it is, you have to worry about pressure when you do go deep on these. Exploration of this is exactly what I was talking about all day yesterday in ways of exploring this. We have an amazing group of people meeting at IDP right now that are world leaders in trying to understand what happens in the seafloor right now. Amazing group of people are sitting out there, right, international group. But as deep as you go, it depends on the amount of organic matter. I showed you guys that core tube from St. George Island that had my hand in it, that had arrows at the top and had a little arrow going to nitrogen, an arrow going to iron, an arrow going to sulfate and whatnot, and that bit. That distribution, how deep that profile goes, is very, very, very closely related to the amount of organic matter. The more organic matter, the more shallow those gradients happen. In a salt marsh, you can go from aerobic to anaerobic in millimeters. So you have burned up all the oxygen and most of the nitrate in the first few millimeters of sediment in a salt marsh. You go out to the sandy sediment environment, and you're talking about a meter, maybe a little deeper, like offshore, a little bit offshore, you can get oxygen penetration of about a meter. Where we're going in some of these environments out, way out, middle of nowhere ocean type bit, we've set records now of seeing oxygen penetration in the oceans that go down to 100 meters. And that's because there is just ridiculously low amounts of activity in those environments. And so more organic matter, the faster everything is burned through, the more respiration. The more you burn up the oxygen, the more you burn up the nitrogen, the more you burn up the iron and whatnot. Less organic matter, the oxygen penetrates a lot deeper. And so that's where you get the spread out of the life, you get spread out of those different lineages, you get spread out of all the things we've been talking about. It's based a lot on this. And the book throws out some numbers here about the amount of fresh water. Before we get into fresh water, I've never finished this stuff yet. Let's take a 5-minute break. Before we jump into fresh water.